In the name of Allah, we will discuss today the brain. What you are looking into? Yes, sure, the bulbs which are lit, right? So we have to discuss today the most complex system of your body called CNS, right? And as you can see in the picture, similar to that, we have got a network of many billions of nerve cells called neurons. And the smaller sizes bulbs you can see concentrate that they could be relate with that of the glial cells, which are supporting in their nature, and you get hundred of interconnection among them, right? So many neurons are supposed to integrate, and this complex system has to process various information and generate various responses. So come towards the actual picture, right? We talk about a lot that the neurons fills your brain. What they have to do? They have to respond the external environment stimuli, and that stimuli alters all the ionic gradient across their plasma membrane, and they have to produce what? They have to create action potential, and in this way they are a part of the excitability or irritability, right? And as a result they produce response in your body. So let's first concentrate the structure of the neuron. Here you can see the cell body, which is having central nucleus and the nucleolus, right? And in the cell body or perikaryon or soma, you have got that there are the chromophilic filled granules called nacelle substance. What are they? They are the rough endoplasmic reticulum studded with the ribosomes. They are supposed to produce proteins, right? Various neurotransmitter could be a part of it. And then you can visualize from one end of this neuron, there could be the initial part called as axon helix. From here, the axons, they are coming down, and you can find the neurofibril inside them. In the axolemma and axoplasm is there. And then the axon are being covered, wrapped by the myelin sheath and with the interval called node of Ranvier, right? And another important thing, you can see that from the cell body, various processes you can visualize. These are the dendrites, right? Now you can concentrate that this one neuron is synapsing or communicating in a sense with the other neuron. And the place where that happens is the synaptic knobs, right? Here they have to secrete their neurotransmitter, then there is a synaptic cleft, and then comes the next neuron, right? Now this is the brain, the picture you are looking into. Here you can see so many convolutions produced by various sulci and gyri. And our topic today would be the cerebral cortex, right? And we will discuss its various cells and the layers, right? Now you can see that there is a brain being covered by the dura mater with its two layers. Then comes arachnoid and pia mater. Now we have to see the cut section of the brain, right? Here in the cut section, the outer cerebral cortex is made up of your gray matter, the seat of intelligence. The central lighter area is white matter, and it is having buried, deep-seated islands of various nuclei. The basal nuclei made up of gray matter, right? Now see in this picture that your gray matter is showing beautiful arrangement of various neurons and the processes which are coming from their cell bodies. And to learn them, we have to move towards other diagram, right? Among these layers of the gray matter, these cells are divided into pyramidal cells and non-pyramidal cells. So let's see the detail of the pyramidal cells. In this picture, you can observe a neuron that is a pyramidal cells. This is of multipolar variety, right? It has got triangular cell body or flask shaped cell body, right? And from its cell body or perikaryon, you can observe that there is an apical larger single dendrite running upwards to end in the most superficial layer of the cortex. In addition to this, a main dendrite, we expect 4 or some more branches or dendrites. They are coming from the base of the perikaryon or cell body of the neuron. Now you can concentrate that there is an axon emerging from the center of the base and is running downwards in the white matter. And there it has to form a projection fiber or an association fiber. Right. After the main description, we should know that pyramidal cells could be classified according to the height of their cell bodies, right? And they could be labeled as small, medium and large, right? And generally their size varies from 10 to 50 micron, right? But in some internal layer of the primary motor area, the pyramidal cells become so giant that they are forming their bed cells, okay? And their cell bodies almost measure up to 100 micrometer in height, and few of them could be found in the sensory area, right? After the pyramidal cells, right, you have to look into some non-pyramidal cells which reside in your gray matter layer, right? And you can see that there are various granule cells and there are some horizontal cells over there, and you find some Martinotti cells are there and few fusiform cells are there, and most of them are interneurons. Granule cells, according to their shape, could be called as stellate cells, right? Now we will move towards their individual description, right? Look into the picture. There is a granule cell. They have got small polygonal cell body or pericarya, right? And due to their shape, they are also called as stellate cell. And they have to give rise so many small dendrites, you would say. They have to pass in various directions, we would expect, right? And due to this, they are appearing as a starry shaped cell body. And we also see that they are giving rise to short axon, and they could be showing the ramification or rebranching, right? And what expect? We expect from them that most of the extrinsic inputs coming towards the cerebral cortex are from the thalamic nucleus, right? Thalamic nuclei are giving these extrinsic input towards your cerebral cortex through these granule cells, and these all information will terminate on the dendrites of the granule cells, right? Then we have to mention the horizontal cell. These horizontal cells of Cajal, as you can see, are horizontally lying, fusiformal body, horizontally oriented, and they occupy your superficial cortical layers, right? They have got short dendrites, you can see on the right side, and a longer axons. They have to run parallel to the surface of your cerebral cortex, and they will make various connection or contact with the dendrites of pyramidal cells, which we discussed earlier, right? After that, now we have to see the Martinotti cells, which are again non-pyramidal one. They could be small multipolar neurons, you would say, looking like cuboidal cells. They are found where? They are located in the deeper layer of your cerebral cortex. They are again showing the short dendrites but long axon that ascends towards the cortical surface because they want to end in the more superficial layer, right? And they in this form reach the outermost layer of your cerebral cortex. Now the fusiform cells, you can see that their cell body is spindle shaped, right? And they are oriented at right angles to the surface of your cerebral cortex, right? And their axon, you can see, is arising from the side of the perikaryon or cell body in its middle part, right? And descends downwards into white matter. And you see that this cell body is giving rise to various branches called dendrites, and they have to extend into the deeper and rather more superficial layers of the cerebral cortex. Now, after discussing various cell details in your cerebral cortex, we have to see that your white matter or cerebral cortex is divided into 6 horizontally arranged layers, right? And this section is taken after cutting perpendicularly to the cortical surface. So we can see from superficial towards deep side the name of these 6 layers. The outermost layer is the molecular layer. Next to it is the external granular layer. Then you find the external pyramidal layer. Then you get into the internal granular layer, and then you find the internal pyramidal layer. And lastly but not the least is the multiform layer of the cerebral cortex, right? Now again concentrate this beautiful picture that is showing the 6 layers. The layer number 1, which we mentioned, the molecular layer, what it carries. Basically this layer is showing the cell processes of the underlying neurons. What processes? The dendrites and axons. So this layer is also called as plexiform layer. And when we cut it, the transverse section will be showing the cell processes, and they will give the punctate or molecular appearance to this layer. So the molecular layer is a synaptic zone of your cerebral cortex, right? And what type of cell we expect here? There could be the neuroglial cells, supporting cells, and we can expect some horizontal cells that are present in layer 1, right? And in the next layer, which is the second layer, is called as external granular layer. That is filled with so many, or you would say closely packed granule cells or stellate cells, and they could be having the large number of smaller size pyramidal cells, right? And then after that we enter into the third layer. Third layer is the external pyramidal layer. As the name is showing that it is mainly consisting of pyramidal cells, but of which size? The medium sized pyramidal cells. And their you would say size increases from the external to the internal border of the layer, as you can see in the picture. And what other cells we expect here? Some granule and Martinotti cells could be a part of this third layer. The layer number 4, which is called as internal granular layer, right? This layer, if you concentrate, is having closely packed granule cells, right? Pyramidal cells are also there. Neuroglia, you can expect protoplasmic astrocytes, which are a astrocyte of gray matter, and you expect microglia and oligodendrocytes. And what else? In this layer, you can expect some horizontally arranged nerve fibers that could be forming the outer band of Bularger, right? I will show you some other diagram to make you clear this band. Now concentrate on the layer number 5, that is called as internal pyramidal layer. This contains the larger size pyramidal cells. You can see that. In the cortex of the primary motor area of your frontal lobe, it consists of the giant pyramidal cells, right? Which we named as Betz cells, B-E-T-Z, right? And what else you find here in addition to pyramidal cells? The cells could be fusiform cells, granule cells, and Martinotti. And they also contain horizontally running nerve fibers, and they will also form an inner band of Bularger, right? And then concentrate on the last but not the least, the 6th layer, which is multiform. The name is showing that here cells could be of various shapes, right? And we can name them that fusiform cells, pyramidal cells, stellate or granule cells, and Martinotti cells, and few neuroglia are a part of that 6th layer, right? In this picture of the laminated layers of these gray matter or cerebral cortex, from top to bottom, you can see various neurons are making synapses with each other. Among them, you can find stellate cell, pyramidal cells, and other cells like Martinotti and some neuroglial cells. And you find that top layer is rather showing so many processes, and when they are cut, they appear as molecules or dots. That's why this layer is called as molecular layer, right? Now in this diagram, I want to show you the band of Bularger. Here you find these horizontal lines which are present in internal granular layer, so forming outer band of Bularger. And there is another layer which is having the inner band of Bularger. This is internal pyramidal layer or layer number 5, and you find here larger pyramidal neurons called bed cells, right? And in this last layer or 6th layer, you can find polymorphic or different shapes of neurons and spindle neurons over there. We mentioned earlier that this 6th layer is multiform layer. And what else? On its lower part, you find white matter, and you can see so many axons running in this direction and are making connection with the processes of the neurons above, right? Now here in the white matter, they have to form various axons collection called as tracks. In this picture on the top, you can see the schematic representation of the 6 layers and the location of various cells. The topmost layer is addressing the horizontal cells along with its processes. The next layer is showing you the stellate cells, and then the consecutive layers are showing Martinotti, and again granular cells be a part of it, and you find the arrangement of the fusiform cells, that how they are lying, and pyramidal cells are also there, and the smaller size pyramidal cells are in the superficial layers and the deeper layer addressing the larger pyramidal cells. And in the lower picture you find the histological H&E stain cerebral cortex, in which the numbers are given to the layers, and you can see there is no clear-cut line or demarcation among these layers, right? Only when the electron microscope is supposed to observe the tissue, the presence of different cells with their different sizes and different location will address that a person is in first layer or is looking into the second, third or fourth, right? So concentrate on the histological view as well. You can see here the Martinotti cells, the pyramidal shaped cells are there, right? These are the picture again to show you that how neurons appear with their cell bodies, axons and dendrites, and bed cells are there. And this stain again you can find the neurons with their processes, right? This color scheme is also showing you the 6 layers. The top one is the molecular. So if you want to memorize, you can learn the last and the first layer start with the letter M, molecular and the multiform, and the second and the third starting with the letter E, external, right? And then next 2 layers are starting with the letter I, the internal. So easy to memorize them, that molecular layer is the first layer and multiform is the last 6th layer. Then the 2 layers are consecutively external, granular and pyramidal respectively. Then comes the word internal, the granular and pyramidal respectively. Now it becomes easy to you to learn the layers, right? Here in this picture we want to see the morphological classification of the neurons according to the number of their processes. So you can see the unipolar and pseudo-unipolar, the bipolar and the multipolar. So first concentrate on the unipolar. That unipolar neuron, which is a sensory neuron, previously may be called as pseudo-unipolar. What they show, they are showing one cell process, right? And in the pseudo-unipolar, what is going to happen? The single process is going to divide into 2 branches. One of it will behave as dendrites and the other will run towards CNS, you would say as an axon, right? And they form or resemble to a letter of T, right? And in this way they do their function. The unipolar neuron, where it is present? It is present in the dorsal root ganglia or spinal nerve, if you remember, and the other place is sensory ganglia of your cranial nerves, right? And what about the bipolar? Bipolar, as the name shows, that this cell bears a single axon and a single dendrite, right? And they are present on the opposite pole of the spindle-shaped cell, right? Cell body, you would say. And these neurons you find where? Again, they are sensory, and you get them in the cochlear and vestibular ganglia related to your ear, right? And you find them in the retina of your eye, right? And then in the olfactory epithelium. So these bipolar neurons are related to your eye, nose and ear, right? That will be easy for you to learn. Now, multipolar neuron. What they are looking like? They are showing a single axon and a number of dendrites, right? Arising directly closer to that of your cell body. Most body neurons belong to this multipolar variety. Where they are found? They are located in your cerebral cortex as pyramidal cells, right? And in the cerebellar cortex as a Purkinje cells, right? And they are also present as a motor cell in the ventral or anterior horn of the spinal cord. So that is enough for you for today to learn the morphological classification. But if we classify the neurons functionally, so we can call them as sensory, motor and interneuron, right? Among them, sensory neurons are those that receive stimuli from the environment or from the tissue or body, and they carry this information towards your CNS, right? And motor neuron, as we mentioned, variety could be that multipolar, and what they do, they have to convey impulses from CNS or ganglia to your effector cells like gland cells or muscles, or to some other neuron. And what's the third type of functional classification of neurons include? These are interneurons, and where they are located? They are basically present in your CNS, and they form a communicating and integrating circuit, right? In between, if you reconsider spinal cord, anterior and posterior commissure rather, you get that the sensory and motor neurons, they communicate through various interneurons. And almost we expect 99% of all neurons belong to this variety. I think this is enough for today. Now we have to learn about the classification of the neuron according to the length of their axons. Previous two classifications we have already learned. So about the length of axon, we divide it into Golgi type 1 neuron and Golgi type 2 neurons, right? In this picture, if you concentrate that the Golgi type 1 neurons, this cell, or you would say I would have to mark them rather, their cell bodies that lies in the gray matter are having very long axons, right? And these axons leave the gray matter running through the white matter and are supposed to form a larger nerve called peripheral nerve. You can see here the formation of spinal nerve. And these types of neurons are found in anterior horns of your spinal cord. And now I have to show you another picture where they also lie. Yeah, this is a picture of the cerebral cortex. Definitely the schematic look. We can see the neurons. The name could be anyone, fusiform, stellate or anyone, right? But we have to concentrate those neurons. You now can see the pyramidal cells, right? These are the examples of Golgi type 1 neuron. What they have to, these varieties, Golgi type 1 neuron, they have many dendrites, as you can see, running upwards superficially, and a very long axon. So they are supposed to leave the gray matter and then running, you can see, downwards through the white matter, and they have to reach the other regions of the CNS, right? So this is about the Golgi type 1 neuron. They have got longer axon. This is one of the way that we divide them into different classes, right? Now here again, what we are supposed to talk about, we are looking into the schematic diagram of the cerebellum, right? Here type 2 neurons, Golgi type 2 neurons are supposed to be discussed. What happens? That they have got many branching, you would say, the dendrites, right? But relatively you can see the smaller axons, right? And what's special about them that they do not even leave the part of the gray matter in which their perikaryon or cell body of the neuron lies, right? They are so numerous, and you find where? You find them in your cerebellum, in the cerebral cortex as well, and usually they are interneuron, and the function of them could be inhibitory. So that was about the classification of neuron according to the length of the neurons. Thank you.